The Hardy-Weinberg Law is the rudimentary law of population genetics. Hardy and Weinberg demonstrated that in a large random mating population, in the absence of factors that change the allele frequencies, strictly speaking, with no selection, mutation, or migration agencies, the allele frequencies and the genotype frequencies are constant from generation to generation. This population is said to be in Hardy-Weinberg equilibrium. In the population under Hardy-Weinberg equilibrium, offspring genotype frequencies are simple functions of allele frequencies of the parent generation. In order to demonstrate Hardy-Weinberg law, let's consider a population that will randomly be mated for 50 discrete generations. In every generation of this stochastic simulation, we have 100,000 diploid individuals, roughly a large population, to nullify the effects of sampling. Consider an autosomal locus with two different alleles at the locus, A1 and A2. The initial frequency for A1 is 0.3. I will explain the concept of allele frequency in a few minutes. There are no selection, mutation, or migration, or concisely, systematic agents that change allele frequency in the population. Okay, now let's describe the genetic constitution of the generated population. There are three types of genotypes: A1A1, A1A2, A2A2. Numbers show how many of each genotype there are in the population in each generation. The proportion of individuals that belong to each genotype is called genotype frequency. In the base generation, the genotype frequency calculated by division of the observed number of individuals in each group by the total number of individuals, as shown in top equation. For instance, 14,201 of total individuals have A1A1 genotype. The genotype frequency for A1A1 is 0.14 or 14%, denoted by capital P. Following the same procedure, genotype frequency for A1A2 is 0.32 or 32%, denoted by capital H, and genotype frequency for A2A2 is 0.54 or 54%, denoted by capital Q. The frequencies of all the genotypes together will add up to 1 or 100%. The allele frequencies can be calculated based on the genotype frequencies. Let's consider the top equations. The first equation shows the relation between A1 allele frequency and the genotype frequencies. The interpretation of the equation in plain English is shown in the second equation. Based upon that, each individual contains two alleles, and in this population, in total we have 200,000 alleles in every generation. Each A1A1 individual contains two A1 alleles, and each A1A2 contains one A1 allele. The frequency of A1 is 0.3 or 30%. In a similar method, we can calculate the frequency of A2. Remember, the sum of P and Q is equal unity. As depicted in the graph, although the allele frequencies are constant from generation to generation, in base generation, we are not able to predict genotype frequencies from initial allele frequencies. However, after one generation of random mating, the population arrived at Hardy-Weinberg equilibrium. In this situation, we are able to predict the genotype frequencies of the current generation from allele frequencies of the previous generation. Hence, the genotype frequency of A1 equals P squared, the genotype frequency of A1 equals 2PQ, and the genotype frequency of A2 equals Q squared. As you can see, our predictions are a good approximation of what we observed. Also, we are able to calculate the number of individuals in each group. Let's run the simulation again with the same parameters. Also, I want to change the initial allele frequency and run the simulation one more time. In sum, as long as the population continues to reproduce under Hardy-Weinberg conditions, the allele frequencies and the genotype frequencies remain constant from generation to generation. I'm Mohammad Sahebhonar, and thank you for watching this video.